Hi everyone, welcome to the final segment of Chapter 17, where we go from RNA to amino acid sequence, that which we call translation. Really nice moving GIF here for you. And again, with the idea that, if you need additional instruction or you need additional context, I can help you find other sources to help make it more understandable for you. And so why this topic is such a big deal is that ribosomes and the genes that encode ribosomes are among the most conserved genes in all of life. And so if we can find a drug that is specific for, say, a bacterial ribosome, like some of these are, then we have a really effective antibiotic that has little or manageable side effects in other organisms that are more desirable, like people or pets and so forth. So it's a very powerful target for certain types of antibiotics. Secondly, it may also be a good way to target cancer cells. Cancer cells are generally rapidly dividing, usually more metabolically active than other non-cancer cells. And dividing and rapidly active cells are more susceptible to chemotherapeutics. They would have more of a need for synthesis of proteins. And so if you can block ribosomal function in cancer cells, you would have a very powerful anti-cancer drug. So when we go through this section, which is 17.4 in your book, we're going to go through this sort of sequentially. We're going to talk about the pieces, namely things like the tRNA, and here's one coming in here, the protein subunits, and then we'll get into the sequential steps, how this sliding occurs, and what's going on as a new tRNA enters and shifts from A to P to E. So we're going to get started with those tRNAs. tRNAs are what allow amino acids to be brought into the ribosome and properly transferred in sequence onto the growing polypeptide. They recognize the codons in the mRNA. They ensure proper amino acids for the codon that is present in the A site there. We're going to start with transfer RNA, a really cool little molecule. It's a single strand of RNA, but it has these internal complementary sequences that create this sort of clover leaf-like structure, where on one end you have what's known as the anti-codon. This is a complementary sequence to codons in the mRNA. And you have the amino acid attached here, which will eventually be transferred off when the tRNA has entered and bound and interacted with the mRNA in the ribosome. There's just several different kinds of pictures of these tRNAs, and the basic function that's going on in what is a fairly complex process. We're going to dial it down a little bit, but we do need to understand the basic steps of what's going on. So tRNAs are not identical. In fact, they have some very important differences. First of all, they have a unique anti-codon that is complementary for a specific codon in mRNA. Like for instance, the anti-codon for this methionine amino acid is UAC, which allows it to base pair if transiently with AUG start codon in mRNA. So each mRNA is carrying a unique or a specific amino acid, and each anticodon is unique and will bring amino acids that correspond to the codons in the mRNA. Here's just a zoom in of the tRNA sequence. And what you see here is you see the letters here, and these are conserved across most or all of the tRNAs. Where you see stars are where they differ in the different amino acid tRNAs. The most important one, though, is going to be in the anticodon. They have letters here, but these anticodons vary. But these stars represent different letters in different tRNAs, which means that that's where some of the variability is as well. They definitely vary at the anticodon, but they can also vary at these others. But with the ones with the dedicated letters here, that generally means those are conserved across the different tRNAs. The bond angles and so forth, this two-dimensional structure is merely a caricature. It's actually more of an L shape. When you see the complements and you include the torsion and the bond angles of the phosphodiester bonds in the RNA, you get this sort of L-shaped structure where the anticodon is down here and the amino acid is up here. A really nice zoom in from your book, and you can see the internal hydrogen bonds in the different areas here, and they're starting to see this sort of a kind of a double helical kind of thing going on, but because there are unpaired areas, the molecule swivels around and it eventually forms this sort of L shape. And that's why we depict it this way in cartoons. There's no need to depict all these different nucleotides. What we're most concerned with is the anticodon in the amino acid attachment site. I'm sure it's easy to imagine that if we didn't have accurate translation, that it would quickly spiral into non-functional proteins. If we had any old amino acid coming in, despite whatever the codon says, that would change certainly the primary structure of the protein and almost certainly the secondary, tertiary, and quaternary structures, leading to a rapid loss of function. So we have two major mechanistic aspects of tRNA that allow for accurate translation. The first is we have to have a correct match between the tRNA and the amino acid for which the tRNA corresponds to in terms of codon, anticodon. And this is done by a special tRNA synthetase that links that correct amino acid to the correct tRNA. The second thing that we have is we've got to have the right matching here. Now, the first and the second letters are super crucial. Those have to be 100% correct. But the third one over here can change around, and that's why when you look at those charts, you'll often see more than one letter in the third position. That's because of something that Francis Crick, a hypothesis that Francis Crick came up with, and that's due to what we call what they call a wobble. This anticodon loop isn't flat like this. It isn't perfectly linear like this. Unlike maybe the mRNA, it's not as linear, and so with that roundedness and with the sort of three-dimensional aspect of this, the fitting here of a nucleotide has a little more breathing room. It's not as precise, and so it can change and thus lead to a tRNA fitting in sort of more than one codon. Like for instance, if this is a G here, this third letter could be a C or a U, and that G would still fit in there. We've done some atomic measurements of this, and that's been corroborated in the physical data. So that wobble is why you see more than one letter in the third space of a codon, because some of these tRNAs can bind to more than one codon by virtue of that sort of three-dimensional interaction. Now moving on to the next molecule that's involved, that would be the ribosome. Ribosome is composed of two subunits, a large and a small, and some ribosomal RNA, sort of a semi-catalytic form of RNA. Again, one of the most highly conserved molecules in all of life. That said, there are some important differences, and so there are antibiotics, particularly some of the mycins, that will target bacterial ribosomes. It says without harming eukaryotic. It just depends on how much dose you're talking about. There are, in fact, some mycin drugs that are used in cancer chemotherapeutics that inhibit human ribosomes as well. So here we have the protein subunits of the ribosome. Just a word of note, we would never see these together like this without other molecules involved. They don't associate this way. They only associate after the process has begun, and we'll get into that detail a little bit later when we talk about the steps. In any case, what we have here is we have the large protein subunit. The large subunit, this is where the tRNAs are going to come in and interact, and we're going to have the peptide being made. We also have the small subunit, which is sort of acting like a guide for the mRNA. The mRNA comes in and moves into this, what looks like a hole here, but it actually becomes it's more like a hot dog bun, in that the RNA will kind of slide through there and have some interface with the large subunit in the tRNAs. We have the A site. This is the aminoacyl tRNA binding site, and this just means a tRNA with an amino acid attached to it. This is where those tRNAs come in. Then we have the peptidyl tRNA binding site. This is where we link the amino acids, and this is an exit tunnel for the peptide coming out of here. And then thirdly, over here on the left, we have the E site. So in sequence, A, P, and E. E standing for exit. So this is where the amino acids come in with the tRNA. This is where the peptide is synthesized. And this is where the tRNA without the amino acid exits. And so I have this written out here for you when I just described in the previous slide. Here's that amino acid tRNA coming into the A site. And then what happens is it gets pushed over. Actually, what happens is the ribosome shifts, and we transfer that amino acid onto the peptide. And the peptide grows out of that exit tunnel right there. And then the E site is where the now empty tRNA leaves the large subunit. We have more pieces of the puzzle here. Here's an as of yet unattached amino acid in the A site, and we see that it has the proper complementary anticodon for the codon. What's going to happen is we're going to shift this over, and in that process transfer this peptide onto this one. This one becomes empty and will move into the E and then be discharged. And like RNA synthesis and like transcribing, we're going to have a three-stage process here: initiation, elongation, and termination. So that's one thing that translation has in common with RNA synthesis. All three require other proteins, and we call these proteins factors sometimes. A lot of times the word factor is used if you haven't definitively identified a protein, and then that term sticks. It gets a little bit annoying after a while, but the truth is, with 30,000 or more different genes, each encoding proteins, some encoding more than one kind of protein, names become a shortage. So there are factors that aid in this translation process. So we're going to simplify it a little bit just so that we can get a start on this. We initiate. What we're going to have is we're going to have the mRNA, we're going to have the tRNA with the first amino acid, and it says 2 ribosomal subunits, but the truth is it's really just the small ribosomal subunit. The big one's available, but it's not bound yet. So we have this small subunit, we have the mRNA and that special initiator tRNA. That's what the tRNA with the first amino acid is. The initiator tRNA is always methionine. Methionine, M-E-T-H-I-O, methionine. That tRNA is the initiator. So every protein, when it is first translated, has a methionine codon in it. So with that initiator tRNA, the small subunit with that initiator, or with the mRNA, excuse me. So we have the mRNA, we have the initiator tRNA, and we have the small subunit, and they're all sliding along the mRNA until it finds a 3-codon sequence, in this case AUG. AUG in the RNA is the start codon. That's for methionine. Methionine has an anticodon to AUG. Can you guess what that is? That would be UAC. In any case, it slides along. That's why sometimes when we do scans of genetic material and we find a start codon like that, we call it an open reading frame. We know that it's possible that a protein could be made from it, but we haven't yet established whether that's an actual functioning gene or not. Now, there are some initiating factors, just other proteins that help bring that large subunit together, help like landing a blimp at an airport or something. You got to bring the big subunit along. This is going to move quickly, so I'm going to let it play once. Small subunit. This would slide along until we found this codon, and then the large would bind. So again, we would have the mRNA, we would have the methionine tRNA in the small subunit, and they would slide along until it found that AUG, and then we would bring that large subunit in. And you can see that the large subunit puts it right into the P site. It's not showing the E site here, but we have an open A site for the next amino acid. So here it is from your book. Small subunit, mRNA with the initiator tRNA, slides along till we find that AUG. And there's that anticodon I mentioned, UAC. In any case, once that has been found, we bring in the large subunit. But we can't do that by itself. We can't get there without a little bit of help from energy. And the energy of translation, in some ways, this is one of the reasons why I want you to stop always talking about ATP as the sole energy, power, or whatever, that sort of a thing. Because other nucleotides can and do provide energy for certain processes, GTP being the foremost of those. So if you were to have a ranked list, you would see ATP at the top and then GTP would be second, a distant second. Nevertheless, though, in order to get that large subunit to bind, we have to hydrolyze a molecule of GTP. And when we do that, we get the binding, and now we have the whole initiation complex in place. Small subunit, mRNA, methionine tRNA, and the large subunit. And we're in the P site. Once we've got that methionine in there, we can begin elongation. And so now the next amino acyl tRNA will come in with the proper anticodon. And what we will do is we will add that methionine. We will shift it from its original tRNA onto the proline. Okay? So we're adding the existing one on top of the amino acid that is coming in. We are reading in 5' to 3' direction. That's a minor detail. But we're always, and the key point here is that when we are synthesizing DNA and we are synthesizing RNA, we are reading 3 to 5, synthesizing 5 to 3. But in the case of translation, we are reading 5 to 3. So that's a major difference there. We're reading 3 to 5 in the nucleic acids, but we're translating reading 5 to 3. And like before, we're using GTP for energy. So every time we shift over one codon, we have to hydrolyze 2 GTP. I'm guessing that one of the hydrolysis is to shift that peptide over onto the new incoming amino acid. And the second GTP is moving the complex relative to the mRNA. Here's the next step. Here's that incoming. We create the bond and we shift it over. Incoming, create the bond, shift it over. Incoming, create the bond, shift it over. But we're always shifting the peptide over to the incoming amino acid. We're reading 5 to 3. Here's the figure from your book, incoming amino acyl tRNA, that's why we call it the A site. The peptidyl tRNA, it has a peptide on it now, is in the P site. Comes in, we hydrolyze a little bit of GTP to transfer that peptide over to that new amino acid coming in. Hydrolyze a little more GTP. That shifts the whole process over. So what was in the P site is now in the E site and is ejected. That tRNA that was in the A site is now in the P site. So we hydrolyze in order to shift that peptide onto that amino acid, and we hydrolyze in order to shift the ribosome relative to the mRNA over one codon. We've been synthesizing this peptide for a little while. It's time that we terminate. Well, there are special codons. There's more than one. There's 2 or 3, if I recall. In any case, stop codon, when we encounter it with the ribosome, when that stop codon reaches the A side of the ribosome, there is a protein that sort of resembles a tRNA. Call that a release factor. It's a protein, but it has a sort of a 3-dimensional structure that will allow it to fit in the A site and interface with that stop codon. It's a pretty cool molecule. That release factor triggers, we add a little water to the end of the, we actually break it up, that's what we're doing. Add a little water to the end instead of a new amino acid. That causes release of the polypeptide, and the translation assembly then disassociates. Review real quick. We're going to hit that stop codon. Here's the release factor. We release and release. Now I've got to add a little more detail, just a little bit more than that. A little bit more going on here. So here's the picture from your book. The ribosome encounters one of the 3 stop codons. The release factor is now able to bind in the A site. What we're going to do is we're going to add a little water here. That's going to release the free polypeptide. It's going to cause release of that, that now empty tRNA. Going to hydrolyze a little more GTP, a couple more molecules, and that causes release of the large and small subunits from the mRNA. And the process is now concluded. So as we're synthesizing the protein, we're actually trying to fold it and shape it. And it can't do that always by itself. Sometimes it can self-shape, and it's capable of that. But a lot of times we're actually going to alter the shape in order to improve functionality or modulate functionality in some way. So as they are, polypeptides are translated, oftentimes they're being folded. And after they've been completed, they may be targeted. They may have special sequences inside the amino acid sequence that target it for specific locations, maybe out of the cell or in a cell membrane and so forth. And we could spend weeks, we could maybe spend a whole semester on just what we call post-translational modifications. And these can literally occur as the protein is being synthesized. If you take an advanced cell biology class, they have these signal sequences that cause, as the protein is being synthesized, to enter the membrane and then cross the membrane again. It's a pretty cool mechanism, but we don't have time for that. But again, these things all occur after the primary sequence of amino acids have been created. Translational modifications, as I said, include folding, include targeting, include covalent modification and or cleavage. So we can cut these things, we can stick them in the membrane, we can send them to the Golgi for additional modification and transport out of the cell. All of these things are considered post-translational modifications. And if a protein isn't folded properly, there are actually proteins that will recognize misfolding and destroy the proteins that are misfolded. There are diseases where so many misfolded proteins are created that the destruction apparatus gets overwhelmed and the cell ends up getting inflamed and or dies of necrosis. Now we've completed the protein synthesis process. We've had initiation, elongation, termination, and then post-translational modification. But what about when things aren't correct, or when we have what we call mutations in the genetic sequence? Those mutations, if they are in the right place and are of the right kind, can significantly affect the structure of a protein and its function. So what we mean by mutations are that either overt changes in the letter sequence in the genetic material of either a cell or a virus, or occasionally you will see chemical modifications of a nucleotide that cause it to behave like a nucleotide other than the one that it is. There are some methylation and some other situations that cause a nucleotide to no longer behave chemically like what its letter is. And there are some different kinds of mutations. For instance, we have what's called a point mutation, and I'll show you some pictures of this in a second. Point mutations are where you have a single nucleotide and its complementary nucleotide on the other strand that gets changed. So you get a change on one strand and then you replicate the DNA or something, and that causes the change on the other strand. Change on the one strand that doesn't get fixed. Now it gets incorporated into the genetic sequence, and now you have a point mutation. Point mutations usually aren't that significant. They happen quite a lot. Nevertheless, a single nucleotide in a DNA template can potentially lead to production of an abnormal protein. I'm going to show you an example of that here. The example is one of the most common mutation diseases in all of mankind is sickle cell anemia. It's the most common genetic disorder in the United States. Just one nucleotide gets changed. We go from a T to an A. All right. If that A gets incorporated, when we replicate the DNA, now that'll be a T. And guess what replicates quite a bit? Blood cell precursors. Blood cells replicate a lot. And so if this error gets in there, you have the possibility of generating lots and lots of mutant hemoglobin. So instead of a GAA codon, which is encoding for glutamine, now it's a GUA, and that encodes for a valine, which is really, really hydrophobic. And what happens is, under oxidative stress, the sickle cell hemoglobin will begin polymerizing and cause long stretches of the cell as these hemoglobins polymerize. And so that one valine, when blood gets a little bit deoxygenated, can cause this hydrophobic interaction, polymerization of hemoglobin, and that polymer stretches out the hemoglobin in such a way that it is no longer this beautiful biconcave shape. And now it can't fit in blood vessels. That's why one of the reasons, and thus it can't carry oxygen to tissues. It's one of the reasons why patients who have significant sickle cell disease often feel a lot of pain, because when tissue doesn't have oxygen, it starts sending out signals, and that includes a pain signal. So in any case, that's a stretched out red blood cell that's been sickled. Point mutation is by far the most common form of what we call a nucleotide pair substitution. We've substituted another pair of nucleotides in that sequence. So a point mutation is one form of nucleotide pair substitution. Then we have a functional term here. This is to say that a silent mutation does not affect the amino acid sequence. Because, maybe it's in the third site on a codon and there's a wobble there. Because of that redundancy in the genetic code, more than one type of codon can encode an amino acid. Most amino acids, with perhaps the exception of methionine and maybe one, I can't think of any others, have more than one codon. So their redundancy and the wobble site mean that a lot of these point mutations and substitutions go silent. They may occur, but they have no impact on the protein. On the other hand, we have what's called a missense mutation, where we get a change, and it could be one or more nucleotides, but now we have a different amino acid, and this has a much more potential to be damaging to the protein. So it's much more potential there because now we have a different amino acid. Sometimes these are also silent because functionally that amino acid is not relevant. It's still a missense mutation, but not the correct amino acid. And then we have nonsense mutations. Now we're getting kind of serious here. Nonsense mutations change what was normally an amino acid codon into a stop codon. So now we've got a truncated protein because it stopped early. It's not the full length that it should be, almost always leading to a protein that's not functional. Lots of diseases out there have nonsense mutations, where a stop codon is inserted where it shouldn't be. Here is an example of a missense mutation. Here's our wild type DNA strand and the coding sequence, okay? And then we get our mRNA. Here's our methionine, and we've got codons going on. We stop, and it's a pretty short protein. They're usually not that long. They're usually 20, 30, 50, 100, 1,000 amino acids long. So we have a substitution. This is a point mutation. But functionally, it is missense. So it's a point mutation because we've only changed one point. But instead of a glycine, we now have a serine here. And so this is a point mutation, a substitution, that leads to a missense. So it's a missense mutation as well, functionally missense. If this were something like perhaps here, that may be a wobble and it would be a silent mutation. Now we have a glycine to serine, and that by definition means missense. That same DNA sequence, only this time let's incorporate a point mutation, again, substitution, right here. And now we have, instead of an AAG, we now have a UAG. And instead of finishing a glycine and stopping, we now have a stop here. And now we only have the methionine. We don't have any other of the rest of the peptide. So functionally nonsense, but it is a point mutation substitution. Substitutions, and now let's look at insertions and deletions. That's where we're adding or subtracting nucleotides. So we're either adding 1, 2, or 3 or more nucleotides. That could cause what we call a frame shift, because it could literally lead to multiple missense mutations or stops. So when we shift that 3' frame, we get what we call a frame shift. So let's look right here. Let's say we have this one right here. Here's our wild type. We insert one more one more nucleotide in there, okay? And now instead of the C being there, now we have this ATT, CAC. We have a completely different and almost certainly non-functional protein being produced by this nucleotide sequence. So there you have it. Big chapter, a lot going on. Take it slowly, work on the different steps. Try not to wait until the last minute to get this stuff down. If you do that, you're setting yourself up for trouble. So thank you so much for your time. Good luck. Let me know if you have any questions, and I will do my best to help you as best I can. Thanks again. So long. And so this completes Part C of Chapter 17. I want to thank you for your time and your patience this semester. I know it's been challenging. As always, if you have questions, if there's something not working for you, I'm more than happy to help you master this material in any way that I can assist you. So thanks again, good luck, and I'll be there for you if you need me. Take care. Bye.